Thank you very much, everybody, to join the today's UCI Mind seminar. It's a great pleasure to introduce today's seminar speaker, Dr. Dana Tudorascu. If I pronounce wrong, I'm sorry.
No, it's okay. That's good.
Great. Just a brief bio of the Dr. Tudorascu. She received the bachelor's degree in the mathematics in Romania, a master's degree in the computational mathematics at the Duquesne University in Pittsburgh, I believe it's in the Pittsburgh, and then Ph.D. in the biostatistics at the University of Pittsburgh. During her Ph.D. work, she was also involved in the Amyloid PET imaging university. After she obtained the Ph.D., she worked as a researcher at the Weizmann Center at University of Wisconsin, basically where she utilized the functional MRI and PET imaging for her research. In 2011, she was recruited back to University of Pittsburgh, where she received a PhD and joined as an assistant professor and established her current work research program since then. She is currently a co-lead of the study design and data analysis group in the psychiatry department. And also she is a co-leader of the biostatistics and data management core for Alzheimer's disease biomarker consortium and Down syndrome. This particular consortium, the two of the UCI faculty, Dr. Liz Head and Dr. Mark Mapstone, is also participating as a multi-PI from the UCI site. She's also associate core leader for the data management and statistics core for Alzheimer's Disease Research Center at Pittsburgh, and neuroimaging statistics in Alzheimer's disease group leader. Her research focused on specifically in improving brain tissue classification in presence of the white matter lesions and atrophy in neuroimaging studies of people with age-related pathology or Alzheimer's disease. She has been awarded grant from NiAD and other agencies, one of which focus on the statistical method to improve reproducibility and reduce technical variability in multimodal neuroimaging studies in Alzheimer's disease, which is a very important research field because we cannot do anything without proper statistics and validating the reproducibility. So I'm very excited to hear her talk today. The title of her talk is Clinical and Statistical Challenges in Analyzing Data from Alzheimer's Disease Studies. Thank you very much, Dr. Tudorascu, today's participation. And without any further ado, I will pass it to you.
Thank you so much. I'm very excited to present this work, and thank you so much for the invitation. So I wanted to do this in two parts. So the first part is just about data harmonization, and the second part, because your institution and Liz and Mark are part of the Alzheimer's disease biomarker consortium, I wanted to present some new results of some analysis that I have done recently with parts of that cohort. So this is just, the slide with an overview about what I'm going to talk today. So as I said, it's a two parts, a data harmonization, when I'm going to talk about two methods that are actually, they have been developed, they are existing in the literature, and we just use them for data harmonization. And then I'm going to talk about the Down syndrome application and some of the statistical analysis that I've done, which is, I would call it a little bit non-conventional, and you will know what I mean once I get there. So please feel free to stop me anytime you have a question. I have 29 slides, so I hope we're going to have time to talk about this before. So what I first wanted to start with is that I do collaborate with a lot of people, and I think without them I won't be able to do any work, because all the data and all the questions that I try to develop statistical methods or to use existing methods are because these wonderful collaborators I have such rich data sets. So I work with a lot of people at University of Pittsburgh, and I have 2 graduate students, which I'm very grateful for because they do a lot of work for me. I have collaborators for statistical analysis at Johns Hopkins. I have a longtime collaborator at the University of Wisconsin, Dr. Christian, who's also part of the ABCDS, one of the MPIs, together with Mark and Liz. Most recently, I started working with Dr. DeCarli specifically on data harmonization at the UC Davis, and we'll work closely with a neuroradiologist at University of Maryland, Baltimore, that does a lot of our evaluation of the segmentations and things like that. So as you can see, it doesn't take a village to do neuroimaging analysis. It actually takes a whole country pretty much. So I want to talk about data harmonization. I think data harmonization is kind of a hot topic these days, and that is specifically because the neuroimaging studies, they have been expanding so much in recent years, and we have all sorts of issues that we have to deal with. And why am I saying that? So we are not only collecting data from scanners with different magnet strengths. So we started collecting data on magnets that were 1.5T, and then we moved to the 3T, and then now we are finally moving to 7T. So it is a challenge to combine all these data and analyze them statistically because there are so many differences between this. But it's not only different magnet strength, it's also different manufacturers. So you can have the same magnet strength, but you can have a scanner that is from GE, one is from Philips, one is from Prisma, and you still have variability due to the manufacturer. And then it's not only the manufacturer, but it's the different institutions. You can have different scanners at different institutions, or you can have the same scanner but different institutions, and there is still variability that is due to the population, and it's also from the scanner. So as we have the ABCDS study where we collect data from, Pittsburgh, Wisconsin, Irvine, WashU, Columbia, MGH, Oxford, Cambridge, I'm sorry. So it is so many institutions that when you are trying to analyze this data, it just becomes very, very hard to do that. So why is it hard to combine this data? So first of all, the main differences between these different magnet strength is the fact that they have, for example, the 3T has approximately twice the signal to noise ratio of the 1.5T. So the 3T scans have much improved contrast between the tissues. So what we are interested in towards at the end is basically the gray matter, white matter, or these CSF volumes that we want to look at. And in order to do that, we have to do segmentation. So the higher the magnet strength is, the better the SNR, so the better the contrast. So if you try to do segmentation on a 3T data or on a 7T data, the contrast is going to be better between the tissue types. So you are going to have an advantage of classifying tissue classes, especially at the, where they overlap at the boundaries. So another issue that can interfere with the scan is the PET MRI co-registration. So when we do positron emission tomography, we always co-register with the MRI studies. So what happens is that PET has very low resolution. So in order to extract the anatomical regions, you have to co-register with an MRI. So you can imagine that if you have a longitudinal study and you have some subjects that are scanned on 1.5T and then some subjects that are scanned on 3T, you have to somehow make sure that there is no difference between their scans, and then the co-registration should actually happen the way you think should happen, so that you're able to find these anatomical regions. And then the other issue with the PET imaging is specifically with amyloid imaging, because what we want and what we are after is this classification into amyloid positive and amyloid negative. And we know that, when we're looking at the Alzheimer's disease, so people that are amyloid positives in their 50s or 60s, they have a high probability of having AD later in life. So when you do these co-registrations, sometimes what happens is that because the boundaries and the segmentations are not delineated very well, depending on the signal to noise ratio and on the MRI scan that you're using, some people can end up being classified as amyloid positive when they are actually amyloid negative, or vice versa. And that is because of the boundaries of the segmentation. And then the third and the most important thing is that, MRIs are arbitrary units. So they are measured, their intensities can vary substantially between magnets. Not only between magnets, even if you put a person in the scanner in the morning, then you scan the person again in the afternoon in the same scanner, you can have variability due to this arbitrary units selection. So what we call this in statistics is some sort of a measurement error. So in order to have an accurate measurement of gray matter, white matter, and CSF, we need to eliminate all these sources of variability. So we need to minimize this measurement error. So we usually do that in statistics different ways. So for example, one way to do that is we minimize the measurement error by design. So when we design a statistical study, we look to have subjects that have specific age and have a distribution that is well balanced between the males and the females in the study. Or if you have different centers, you look for populations that is heterogeneous within and homogeneous between the centers. So you want to make them as close as possible, such that whatever differences you have are not differences due to certain sources of variability, but are differences due to medication or whatever else is the outcome that you're measuring. In this case, there is another instrumental error that is, contributing to the variability, and that instrumental error is from the scanner differences. So what we're trying to do is we're trying to minimize this scanner differences and this error, and we want to capture exactly what is happening in the brain that is due to the disease, but not to this scanner variability. I think this is very important, specifically now, because the end goal of some of these studies is, as much as we want to understand the disease, but it's also, pharmaceutical. So we want to give subjects some sort of a medication, and then we want to measure the response to the medication. If we think about let's say aducanumab, right? That's one of the Alzheimer's disease drugs that is being, FDA is evaluating right now. So the outcome measure for the aducanumab was atrophy. So atrophy is measured as gray matter volume shrinkage, over time. So if you are not measuring gray matter volume with accuracy, how do you know that the medication is doing what's supposed to be doing? So I think it's very, very important to account for all these sources of variability such that we can capture, the effect of the disease or the effect of the treatment very accurately. So we've done a very simple experiment. So we were very fortunate here. We had 16 elderly subjects that were scanned on 1.5T and 3T data, and they were scanned 3 months apart. And what we did was, so we wanted to specifically look at them and see what is happening with these subjects. What's the difference between, their 3T scan and 1.5T scan. So being scanned 3 months apart, that means that whatever was happening in their brain, it was not necessarily due to clinical deterioration or to the disease, because the period of time was very short. They didn't have any strokes or anything that would be some sort of an inflammation that happened within that 3 months. And then the other thing that we wanted to investigate, which is some sort of a reproducibility, if you want to think about it that way, was, we use 2 software to look at these outcomes, and specifically gray matter. So we used SPM, statistical parametric mapping, and we used the free surfer. And the gray matter volume was the outcome that we compared. And we looked at others too, but this was one of the most important, since this is actually used, you know, in some of the studies as the most important outcome of interest. And then we look at between scanners within each software, and then we look at within scanner between software differences. So just to have some ideas of how this data look like is that, so for example, this picture here on the right, this is showing in red, this is showing a plot of the gray matter intensity values, and this is from a 3T magnet, and then this is from the 1.5T magnet. This is the same subject, okay? So what is striking is that when I look at this as a statistician, it's like that can't be true. You know, this is, you can't have this kind of a difference between the 3T and 1.5T for the same subject. It's 3 months apart, but that is just scanner differences. They should overlap very nicely if there shouldn't be intensity differences between them, but they are not. So we put these 16 subjects together and we calculate the volumes in free surfer and SPM. So in this table right here at the bottom, you can see the gray matter volume for the 1.5T, and for the 3T is the second line. And the third line is basically the mean differences. So when we calculated the gray matter volumes with the free surfer, the 1.5T was 622 millimeters cubes, and then the other one was 526 milliliters. So the difference between the two scanners in free surfer, between the 16, these are same subjects scanned on 1.5T and 3T, and the difference between their volumes was 96. And the difference in the SPM was 52. So in SPM, we saw smaller scan differences. That was due just to the pre-processing technique. And basically within each scanner and between softwares, we've also seen differences between free surfer and SPM in terms of the volume, like a 57 milliliter difference, and in the 3T we've seen a 91 milliliter difference. So it's not only between scans, and it's also dependent on the software that you are using. And we've done this research before, and we look at the differences in SPM and FSL on young subjects and healthy subjects that were scanned 3 hours apart on the same scanner, and we found the same type of differences. So basically it was very dependent on the software. Here is not only the software, it's also the scanner. So in terms of, you know, this is ICC or some sort of an agreement, if you want to call it like that. So there was no agreement between the 3T and the 1.5T. An ICC of 0.27 means no agreement. And this is 0.72 between SPM 3T and 1.5T, so that was a little higher. However, within scan between software was also low, 0.64, and for the 3T in free surfer was 0.37. So as you can see, there are major sources of variability at play here when we look at data on the same subjects on different magnets. So we looked in the literature of genetics. The field of genetics had a lot of these issues, and they are, I would say, a couple of steps ahead of the neuroimaging. And they have developed a lot of methods for these batch effects. So one of the methods that they had, it was called SUV, and it was developed here, and it's named RAVEL. So a graduate student that was, you know, doing, he was specializing in developing statistical methods for genetics, and then he worked with the brain data, and he said, I've learned all these methods in genetics, why don't I just apply this to brain? So this is how RAVEL was actually born, which is called removal of artificial voxel effect by linear regression. This is an intensity normalization technique, and it's basically it basically has two steps. So you conduct a factor analysis on the control voxels. So what we call the control voxels are voxels in the brain that are supposed to not be affected by the disease. So in the case of Alzheimer's disease, these are the voxels in the CSF, the voxels in the ventricles. So whatever is happening in the ventricles within a certain period of time, it should not be due to Alzheimer's disease, it should just be due to scanner differences. So you do some sort of a factor analysis or a singular value decomposition for the ventricles, you estimate those weights from there, and then you use them in a regression model. At the brain level, that includes some of the covariates that you're interested in, some of the covariates that are biological, such as age and such as gender, and then the direction of these unwanted variations that are estimated from the ventricles as covariates. And the end goal here is to obtain an analysis about the biologically relevant predictors while you account for these sources of unwanted artificial variability. And then, so this is kind of the statistical model that I was talking about. So it has two parts. So for each voxel, so you have a part that is due to the biological variability and a part that is the unknown variability that is estimated from the CSF. So these here are both unknown, and you fit another model in the CSF and you estimate them, and then you plug them back in the RAVEL model. The methodology, as I said, is all existent. The actual code to do this intensity normalization is publicly available on the Neuroconductor website. So it can be very easily implemented. And then the second part was, so the RAVEL is just an intensity normalization. So that will take care of some of the variability of the intensities, but then you still end up with data that is not necessarily harmonized. So we look at the ComBat. So ComBat is, again, has been developed for the genetic field for the same batch effects. And he was the same graduate student that worked at Hopkins. So he first developed RAVEL, and then he took ComBat from the genetics and he applied it to the imaging data. And ComBat is the same type of model. It is the same thing. It's a regression model, but it has, a matrix of covariates of interest, and then it has a multiplicative effect and an additive effect that is accounting for the variability. This is not applied at the voxel level. We are working to do that now, but this was developed for regional level data. It was first developed for the diffusion tensor imaging, and it was applied to MD and FA maps, and then it was applied to cortical thickness. So we use this ComBat for the ROIs. So it is the whole pipeline that we did for data harmonization. It's pretty straightforward. So we basically we start with the raw MRI images, and we derive 4 different outcomes. So the first we take the raw images, and then we apply RAVEL, which is the intensity normalization, and then we apply the free surfer, and that way we get the summary measures or the ROI, and we call this RAVEL normalized measures. So you will see in the next table, we're just calling them RAVEL because we applied intensity normalization. The other way that we have done this is basically we take the raw images, we apply free surfer, and we derive the ROIs. And then on the ROIs, we apply ComBat. So we don't apply RAVEL in this one. So we call this just the ComBat. Then the third way would be to take these raw images and apply intensity normalization. We get the RAVEL normalized images, and then we apply the free surfer. We derive the ROIs. We apply ComBat on these ROIs, which were, RAVEL intensity normalized, and then we derive the measures which we call RAVEL-ComBat, which are actually sort of like we would call harmonized measures, because we apply intensity normalization and then we apply data harmonization. And we compare all of these scenarios with just, outcomes that were derived directly from the raw images. So you take the raw images, you apply the free surfer, and then you get your ROIs. You don't apply any intensity normalization or any data harmonization for them, because you might just have data that is from the same scanner. So you don't need to apply these methodologies. So exactly how you would do a neuroimaging analysis in the presence of, when none of these scanner variability exists, or is just data from one center. So in terms of intensities and some of the results, step by step after applying these pipelines, and this is part of the RAVEL. So in the first row here, you can see that these are the raw intensities after we did the segmentation. So this is the CSF. What is at the bottom here is the 1.5 T scan, and this is the 3 scan, the blue ones, and then this is for the gray matter, this is for the Y matter. So these are intensities, raw intensities, after we've done the segmentation without doing any RAVEL or anything. The first is part of the RAVEL and is called white stripe. So it's some type of normalization within the RAVEL pipeline. So these are making the intensities a little bit closer together, but they are still not overlapping as much as we want them. And this, the last line, is basically the intensities that have been RAVEL normalized. So now they look very similar, the 3T scanner and the 1.5T scanner data. And after we perform all of that, the raw, the RAVEL, the ComBat, and then the RAVEL-ComBat harmonization, we calculated, all the ROIs. So what I'm showing here in this table is the first part is the cortical thickness that was derived for some of these measures that were important in Alzheimer's disease, and some of the volumes. So the volumes are similar to the cortical thickness areas, but we also have amygdala and hippocampus here, which are part of the AD important regions where we see changes in anatomical measurements. So the first column is showing the mean and the 95% confidence interval of the differences in the cortical thickness between these 1.5T and 3T data. So what is shown in blue are differences that are statistically significant in the first column. So we don't do any type of normalization here. So we're just looking at the ROIs differences that were derived from the free surfer. And we see that there are significant differences between 1.5T and 3T in, in entorhinal, left and right, in fusiform, left and right, in the inferior temporal, the middle temporal, the amygdala, and again, for both for left and right. The ones that are in yellow are the ones that are borderline significant, which means that P was equal to 0.5 when we tested the statistical significance. And then the ones in white are the ones that there was no statistical significance between 1.5T and 3T cortical thickness derived measures. So as you can see for the volumes, there were all these that were statistically significant and some of them that were not. Then after applying the RAVEL intensity normalization, we found that some of these measures that were statistically significant that are now in green, they are no longer statistically significant. So these differences between the 1.5T and 3T are actually decreasing. So our goal is to minimize them as much as possible, make them closer to 0. So after we perform RAVEL intensity normalization, it seems that we still have 2 that are statistically significant, and there are some of them that are no longer significant, and in the same with the volumes, but there are still these differences are still high compared to what we started with. Then remember, this is ComBat, so we didn't perform RAVEL, so we just take the raw and we do the segmentation in free surfer we derive the ROIs, and then we apply the ComBat pipeline, which is data harmonization. So now we do have some that are no longer statistically significant and decrease differences. And we still have some in blue that are turning up now, that here they were no significant in the RAVEL, but they are significant now in ComBat, these differences. So, you know, it goes from 0.6 to 0.11. So it is increasing. It's half of the raw difference, but still it's high. And in terms of volumes, these are no longer statistically significant with the exception of inferior temporal. So these differences have been, they are decreasing compared to the raw. So I usually go back to the raw because this is the one that I'm interested in minimizing. So the differences in the 1.5T and 3T data. So for the past, you know, for the last combination that we have done, which is we have performed intensity normalization, and then we have performed data harmonization on the ROIs derived from the intensity normalized images, we've seen that that was basically the best pipeline. So I'm not necessarily looking here for statistical significance, although I only have, so from what I started with in the raw, there is only one difference right now that is statistically significant, and that is in the inferior temporal. The rest of them are green, which means they are no longer statistically significant, and then the white ones that were no longer statistically significant from the beginning. But what is important to see is that we started with a difference of 0.22, 0.24, and now, you know, we have a 0.10 difference after we apply RAVEL-ComBat between a 1.5T and 3T. So here, for example, in the inferior temporal, there was a 0.25 difference in the cortical thickness, and now we have a 0.08. Now imagine that cortical thickness is such a small area, and it's very, very little, and has a lot of variability, and it has to be estimated very accurately. But these differences are diminishing so much compared to what we started. In terms of the volumes, these were higher, but again, when we applied both RAVEL and ComBat, these volume differences have decreased considerably. So from a 0.79, we go to a difference of 0.26. So that is a third of a difference of what we had. You know, amygdala, so now it's 0.4, and hippocampus, it kind of stayed the same. It's no longer significant, but I assume that's due to the variability. In a small sample, you also have higher variability, and that can somehow interfere with the statistical significance. But still, you know, these values that were so high are much, much smaller now, of the you know, order of magnitude smaller compared to the actual differences that we started with. So what we did was also, we wanted to ask a neuroradiologist and say, look, we have done, you know, we have these raw images, we perform segmentation, and we also have applied this intensity normalization after we perform segmentation again. Do we see any difference in terms of, you know, when we look at hippocampus, for example, which is an important area, do we see any differences in the segmentation between the raw and after we've done this intensity normalization and we segmented again? Can you tell us, you know, how would you rate them? So we basically created this 4-point scale rating system where we said, you know, 1 is a poor segmentation, 2, it has some errors, 3 is a good segmentation, and 4 is an excellent segmentation. And then they rated these images and based on for all of these subjects, and these are, you know, for right hemisphere and for left hemisphere, and you know, these are the ones that are rated as being good. I mean, 3 is a good, and 4 is excellent. So very few were excellent, but it seems that raw and RAVEL are kind of like similar in terms of numbers. So in the left hemisphere, it seems that RAVEL is performing worse than the raw, and in the right hemisphere, RAVEL is performing better than the raw. And then if you want to see an image, for example, so this is an image where RAVEL normalized images are performing better, and the raw images that are non-normalized or anything are performing worse. And by worse, what do I mean by that? So the neuroradiologists actually look at the segmentation and overlay them. So one was the one in red is the RAVEL normalized, and the one in green is basically the raw normalization. So it seems like there are some extraneous voxels that are included in the raw in the raw data. And that means that if we perform free surfer on the raw data, there is some stuff in here that does not necessarily belong to the hippocampus, but is labeled as being hippocampus by the free surfer. And there is more, also it's shown up here and it's shown up here. So the idea is that RAVEL was much more accurate in, in the determining the hippocampus segmentation as compared to when we did not do intensity normalization for some of the subjects. But there are, subjects, this is a different subject where RAVEL is actually performing worse. So RAVEL is the raw, and then in green is shown RAVEL is the intensity normalized, and it's the red one, and in green is the one that is correct. So in this case, the green is the raw images. So RAVEL is not segmenting correctly. After we apply RAVEL, free surfer is not segmenting correctly the whole hippocampus. So it seems like it's not including these areas that are part of the hippocampus, but they were not included. So in a different paper, we have actually done this analysis, and we applied RAVEL intensity normalization on the PET data. So, and we also did it for the cortical thickness, but we looked at the NiAD study where we had Pitt data and Wisconsin data. Now the PET scanners were the same. The MR scanners were different. They are the same magnet strength, but one was, I think Wisconsin has a GE and Pittsburgh has a Prisma. And these were, the cortical thickness, we found significant differences between, between the normalized, between the RAVEL and no RAVEL data. But with respect to the PET, we found that it doesn't influence the PET that much. So what I have displayed in this table here is basically looking at the global SUVR values that are determined based on the global SUVR based on the Pittsburgh imaging, Pittsburgh compound B. So this is PiB amyloid binding. And global PiB value for, this is between negative and positive subjects. So we had 30 negative and 10 positive subjects in this study. And we can see that when we didn't perform RAVEL, for example, the global value was 1.1. And when we had used RAVEL intensity normalization for the MRI, then co-register with PET, we had 1.11. And then for the positive subjects, it was a 1.78 for no RAVEL and 1.8 for RAVEL. So when for the anterior ventral striatum, which is one of the areas where amyloid deposits first in the Down syndrome, we see that there was 1.28 when we didn't have RAVEL and 1.31 SUVR value when RAVEL when PET was co-registered with the RAVEL intensity normalization. And in the positives, again, a very small difference, and effect sizes were just a bit different. But what I wanted to tell you is that the differences are small, but we have hard cutoffs for amyloid binding, right? So we consider a subject to be positive if it's 1.5 in the global SUVR, and it's negative if it's less than 1.5. What happens is that when you use an intensity normalization technique, you can have a difference between 1.8 and 1.31. It's just a difference of 0.03. The thing is that that difference can make a subject be positive, classified, being positive with one method and negative with another method, or vice versa. And that is because we have these hard thresholds that we look at. And this is where it can make a difference if you're using an intensity normalization method or no intensity normalization method. And again, it all goes back to how accurate are you delineating those areas. And I think that is $1 million question, because how accurate can we be? And even with the neuroradiological studies, and even with, with our people that are so good at looking at these images, it's very, very hard to think about that there are 3 voxels in these areas that weren't correctly classified, or 5 voxels in that area. So it's such a fine-tuning issue, but it's such an important problem. And I think it's going to be important for many years from now, and it's more prevalent, and it's much more acute in the MRI compared to the PET imaging. So I would say we're a little bit safer with the PET, but not with the MRI. So that kind of concludes the first part of the harmonization. So in summary, I'd like to say that, the results that basically suggest the need for very careful consideration of scanner effects when combining studies from different scanners, different sites. And I've shown that combining an intensity normalization with an ROI harmonization, RAVEL and ComBat, it provides superior results in terms of scanner data harmonization. And for the PET-derived ROI SUVRs, for the amyloid imaging, the differences are smaller, but it can make a higher impact when we classify subjects into positive or negative because of the hard threshold that we have. MRI-derived measures are much more affected by scanner differences, and all the credit for all this harmonization work goes to Mark Bane, who is a graduate student in computer science that is working with me. And we have a paper in preparation and ready to go out with these harmonization results. So I'd like to go now to the Down syndrome application, unless you have any questions about the harmonization. If you do not, I can carry on and we can talk more at the end. So this part is a very straightforward statistical analysis, a little bit unconventional, I would say. And the data that I'm presenting here is part of the Alzheimer's Biomarker Consortium. Originally, the data used here was, it was part of the NiAD study, neurodegeneration in aging Down syndrome, which is now part of ABCDS, and Liz knows all about it. So Liz and Mark at the UCI are the part of the team of the MPI, and Brad Christian at Wisconsin, and Ben Handen at the University of Pittsburgh. So the question of interest was very simple. So we were basically interested to explore the association between amyloid deposition over time and cross-sectional tau measured using AV-1451 in our Down syndrome cohort. The data that we used here was the data that was collected at the University of Pittsburgh and the University of Wisconsin. And PiB amyloid SUVR measures were assessed longitudinally. Some subjects can span up to 8 years of follow-up data. And for the tau, we only had one assessment, and which was sort of like baseline, I would say. The first time they did, an amyloid SUVR, they also did tau, but I think it was 1 year after that because of the radiation thing in the tracer. So the imaging data analysis. So the imaging data was processed using FreeSurfer, using our pipelines. We look at the BRAC ROI. So these are not BRAC. So these are not BRAC stages. These are BRAC regions. And they were summed as previously described in the literature. There is a paper by Scholl, which shows how to get these BRAC regional ROIs. And then we did ComBat to see if there were to basically control for scanner differences in the PET measures, but we didn't find that ComBat was made any difference in this case. So I performed a two-stage analysis. So we basically look at I created one model for amyloid trajectory. So that is the amyloid deposition, which was measured on a span of 8 years for some subjects. And then in the model, we had the time, we had age, We control for age, we control for site. There was a time by site interaction, and then a random slope for time, which is basically each individual which measures how much variability, how much does the trajectory of each individual subject varies compared to the average trajectory. And then we estimated the average trajectory that was computed from the model, and it was used in a non-parametric association with each BRAC ROI, AV-1451. So basically, for each subject, I have a trajectory. I estimate the amyloid trajectory over time. So I take the estimate of the random slope, I take the estimate of the random time, and I multiply that by the amount of years that the subjects are in each individual subject is in the study. So that gives me some sort of an average estimate of amyloid deposition over the period of time that the subject was measured or was observed in this case, right? And then I wanted to correlate that with the AV-1451. So very simple. Instead of correlating at each time point, I take into account the fact that the measurements are correlated over time in a mixed model, and then correlate that with tau, with AV-1451. So the descriptive statistics for the subject is that, so we had 46 subjects. There were 23 PiB negatives and 23 PiB positives, 24 males, and 10 males for the PiB negative, 14 males for the PiB positives. The average age at the measurement of the tau scan was 41 overall, and the PiB negative subjects were a little younger, in the 36 average, and the positives were older at the 47. So the regional BRAC measurements, as you can see, they are higher at the beginning, and they are less tau towards the end, because they catch up with the amyloid deposition towards the later in the BRAC regions. And then the same kind of thing we see for the PiB negatives. So the SUVRs for the BRAC are higher for the region 1 through 4, and then they're kind of decreasing towards the end, because BRAC region 6 is the one where you see tau deposition later. In the positive subjects, the values are higher compared to the negative, but also the same type of trend, which is clinically how it should be. And this is how these trajectories of subject look for amyloid deposition. So you can see the positive subjects. These are the global Down syndrome, the global DS, the striatum. The positivity is based on the striatum, because that is the first area where amyloid deposition hits for the Down syndrome, and they are higher compared to the negative subjects that are down here. And these are the estimated trajectories for these subjects, for each individual subject, from the linear mixed effects model. And then if we look at the distribution of the BRAC regions, you can see the BRAC regions, it's just values are smaller, as we have seen. They are increasing towards, with the first stages of the BRAC deposition. There are a lot of outliers in here. That's why I picked to choose non-parametric analysis. Much more variability in the beginning stages, and lower in the beginning ROIs of the BRAC, and lower in later on. And then these are the correlations. So we looked at the correlations that were adjusted for age and that were non-adjusted for age. And this is specifically for Liz, because we talked about this, what's going to happen with age and how are we going to treat age. Well, the thing is, and it was very interesting, that when we put age in the model and we didn't put age in the model, we did not see any type of change when we adjusted these associations between the amyloid trajectory and the tau. We didn't see any difference in the unadjusted associations compared to the age-adjusted association. So as you can see, the BRAC region 1 correlation with the amyloid trajectory is about 68, 0.68, which is pretty high, and it's the same for unadjusted. And then for region 2, but we have to be careful with region 2, because in region 2, there is choroid plexus and it's non-specific binding. So I was told by my collaborators that know a lot more than me about all this, that region 2 should not be interpreted, or interpreted very carefully, because of that non-specific binding. So it might not be that high. It might just be high because of that deposition in the choroid plexus. And then region 3, 4, and 5, they're much higher. They're 0.86 and 0.83. And then region 6 is a little lower in terms of the associations. So all the analysis was performed in R. We use Neuroconductor, which is a platform for imaging analysis with which we work with our collaborators at Hopkins. It is free. It's available for everybody if anybody wants to use it. And then in summary, we have shown that there is a strong association between the longitudinal amyloid trajectories, deposition, and tau in this Down syndrome population for 2 sites. The association between these trajectories and tau does not change when we control for age, specifically not in these 46 subjects that we looked at. And the region 2 should be carefully interpreted due to this non-specific binding of the tau AV-1451 in the choroid plexus. And these are some of the references, and this is, thank you very much. And that's all I had for today. And please feel free to ask me questions.